Okay, so today we are going to continue on talking about DNA. We're going to go over a few more experiments, just talking about the history of the discovery of DNA. And then we're going to start to go into DNA replication. So we talked about We talked about Watson and Crick last time and how they discovered the double helix form. Now, they won a Nobel Prize in medicine for finding the structure of DNA. However, they kind of were given a lot of assistance, we'll just say. So in a different lab, there was two other scientists, Wilkins and Rosalind Franklin, and they were working with X-ray crystallography, and they were taking a bunch of pictures of a cross-section of DNA. So what they were doing, guys, is they were actually like making DNA into crystal form and then kind of taking pictures of it. It's like, hold on, pose, okay, let me take a picture. So Rosalind Franklin came up with this picture over here, and this would be a kind of like an overlook of DNA looking downward. And they saw this X formation. Now remember, this is DNA pretty much like frozen. It's in crystal form. So in reality, in real life, obviously, it would be like moving around a little bit. But since it's frozen, the X, that represented the double helix. So she pretty much had this picture. She took the picture, and for some reason, it was Watson and Crick that got credit for discovering the double helix. So what happened here, guys, was Wilkins was in the lab, and Watson and Crick were kind of fishing around one day, and Wilkins handed over the picture to them, and they took that picture and kind of ran with it, and that's how they discovered it. Now, Wilkins, Watson, and Crick, they all won the Nobel Prize. But Rosalind Franklin, unfortunately, she did not. Reason being, she actually died very, very, very young because she got cancer. Well, she worked with X-rays, X-ray machines. If you guys have ever had an X-ray done, they put these lead bibs on you to make sure you are not being radiated. And I'm sure that she probably didn't wear those lead uniforms all the time. So that's probably how she got cancer and died, unfortunately. But, you know, if she were there, she probably would have shared that Nobel Prize with the three gentlemen. Okay, so now structure of DNA. So we have two different ends, and we're going to talk about the ends in a little bit, guys. But before we get to that, I just want to mention a few things. Let's talk about bonding. If you remember with bonds, we had hydrogen bonds, we had ionic bonds, we had covalent bonds, and those were the most important ones that we talked about. We also had van der Waals forces and some other things, but the main ones that we use in biology are hydrogen bonds and covalent bonds. And we have three parts to our nucleotide. So here is one nucleotide right here. Okay. We have our phosphate here in yellow, our deoxyribose 5-carbon sugar right here in blue, and then lastly our nitrogen base over here in green. Now here and here, these are going to be our covalent bonds. So what attaches the phosphate to the deoxyribose and the deoxyribose to the nitrogen base? Those are covalent bonds. So they're strong bonds, okay? When we connect one side of our double helix to the other, which is going to be right here in the middle, all right? Now, look at the zigzag I made there. You guys can see it does zigzag, but the ends are parallel to one another with the phosphates and the deoxyribose, okay? Why the middle? Why we have that zigzag there is because it's always going to be two rings on one side and one ring on the other. If the two rings are on the left, the bond's going to shift slightly to the right. And if the two rings are on the right, the bond's going to shift slightly to the left. But those bonds that are going to connect our one side of DNA, in other words, you know, this side here, to that side there, The bonds in between them, these are going to be hydrogen bonds. And if you remember, hydrogen bonds are much weaker than covalent bonds. So if the DNA is going to break, it's not going to break here. It's not going to break here. It is going to break right down the middle, right here on our weak hydrogen bonds. Okay? So I just wanted to mention that before we move on. Also, guys, I'm going to be going over the structure of DNA. And like I said, that 5', that 3', the things you see at the end, we'll talk about those too. Okay, so like I said, guys, let me just draw it out for you. I think it'll be easier this way. Now, the phosphate, I'm going to make an orange. So I'm going to start here, and I'm going to make our phosphate just like that. Okay. Bonds. Here's our bond. And I'm going to make our deoxyribose in this green color. Okay. Now I'm going to draw this bigger, and then I'm going to erase it. But I want to draw the first one bigger for a reason. This is deoxyribose. Now, if you remember when we talked about deoxyribose or ribose, it was a 5-carbon sugar. 5-carbon sugar. That's deoxyribose and ribose. Now let me just label this. I'm going to label our carbons. Okay? This is an O. Every time we come to a point, that is a carbon. So that O does not count as a carbon. Going to the left, I'm sorry, the right, or clockwise of the O, this is carbon 1. This is carbon 2. This is carbon 3. This is carbon 4. And then up here is carbon 5. Okay? Then, attached over here, let me draw our next bond there. Attached over here, we are going to have our nitrogen base. I'm just going to call this one A. Okay? So, after we have, this is one nucleotide. After we have that, I'm going to attach one more, and then I'm going to go over that 5 and that 3, what's going on with all that stuff. So, here's our bond to our next phosphate. And then we are going to make another deoxyribose here. I'm actually just going to make this one a little smaller. All right. And then still we have carbon 1, 2, 3, 4, and 5. And 5. All right. And we will make this next one, make it pink, why not? And we'll say that one is, let's do C for this one. And my bond right there. Okay, so if you notice in DNA, we are going to have a 5 side and a 3 side. And you will see them abbreviated like this and like this. That little guy means prime. So we refer to them as 5' and 3' ends. Now, when you are doing this, guys, what you're doing is you want to look at the carbon. And that carbon is going to help us determine which side is 5' and which side is 3'. So I'm going to give you the long way first, and I'll show you the easy way after that. So I have one nucleotide right here, and I'm just going to draw on it, and then I'm going to erase it. Okay, so there is one nucleotide. So we're only going to be looking at that right now. So pointing up toward the top, and we're just looking at this side of it, pointing upward, you see that number 5 carbon is right there. So the top, this side, is going to be 5'. Look at the other end. If you look at our number 3 carbon, it's facing downward. So we say that this side is 3'. Okay? Another way to determine this, guys, is wherever the phosphate is, if the phosphate's on the top, that's the 5' end. If the phosphate is not on top, okay, that's the 3' end. You're not looking for phosphates really on the bottom, but wherever the phosphate is, that's your 5' end. And you're like, okay, Colm, isn't the top always going to be 5'? No. Watch. So let me do the other side of our DNA here, which I always have trouble drawing this because it's completely opposite of the other one. But I will do my best. Okay. And then we'll make him in this color. And I do the A to bonded to the T there. And if you guys look, I made the A bigger because A and G are the purines, and if you remember, they have 2 rings. C and T are the pyrimidines, and they only have 1 ring. But if you look at this one, if we label our carbons again, so we kind of got to think of this as being almost upside down here, which is slightly difficult for us. But yeah. This would be number 1 here. This would be number 2. This would be number 3. Here's number 4. And then lastly right here is number 5. Okay? And I could draw the other one down here, guys. But if you look at the difference. Just look in this area right here. Whoops. If I can draw it correctly, sorry. Just look at this nucleotide being bonded to this nucleotide right here. This one is one direction. The other one over on the other side is flipped. So it's completely the opposite direction as that one. So if you look here, the 3' one is pointing, or the 3 carbon is pointing up. So this means this side would be our 5'. And then down here would be our 5'. They're called being anti-parallel to one another. So our 5' is always going to be across from the 3'. And our 3' is always going to be across from the 5'. Also, one side always has to be 5', the other side always has to be 3'. And then, I didn't draw them, guys, but you guys would see there'd be, some hydrogen bonds in between there. Okay? Cool. All right, so yeah, that was a good bit, guys. And here's that word, anti-parallel, that I talked about, that they are flipped from one another, the one side to the next. All right. So we already discussed this one. I talked last time about Watson and Crick. They always thought that A was going to be bonded to A, G was going to be bonded to G, but the pairings were not going to be a uniform width. Reason being is if you bond A to A, that's going to be 4 rings going across. If you bond T to T, that's only going to be 2 rings across. At any point, we want 3 rings. We always want a 2 ring bonded to a 1 ring one. And you guys can see here, if we bond 2 purines together, okay, it's too wide, 2 pyrimidines too narrow. We want to always have 1, 2, 3 rings going across. And that was how we got our A is bonded to T, apple in the tree, and then our G was bonded to C, car in the garage. Okay. And there's your bases pairing together. So A bonded to T, if you look here, we have 1, 2 bonds there. And then G bonded to C, we have 1, 2, 3 bonds. So A bonded to T, we have 2 hydrogen bonds. G bonded C, we have 3 hydrogen bonds going across. So there's your difference between those two. Okay, so let's go over the copying mechanism for DNA. And we talked about this last time. Anytime we are going to replicate our DNA, the goal is to make new cells. And when we're going to replicate it, we have to do that before we go into cell division. And our goal is to get 46 chromosomes in each one of our brand new cells. So Watson and Crick started, guys, with trying to determine how DNA is copied in our bodies. And this is one of the ways we were thinking that it is going to get copied. So we have our 2 original strands in that dark blue color. And after we go through DNA replication, it shows on this picture that we get 1 new strand on each of them. So each one of our cells, if this is cell 1, we'll say this is like our DNA for cell 1 here, and this is our DNA for cell 2 here. Each one of our cells is going to have one of the original copies, right there and right there, and each one of them is going to have our new copies, which is the light blue color. All right, so each one gets 1 old, and each one gets 1 new. Oops. Now, that was one of the 3 ways that they thought DNA could have replicated. We already talked about this one, so let me move on to this one. That one I just showed you, that is called the Semiconservative Model. There's also a conservative model and a dispersive model. And let me show you guys a picture of each of them here. So the conservative model, this would be cell 1. Whoops. This would be cell 1 here. This would be cell 2, keeping those colors consistent with the slide we went over before. Conservative says that 1 cell is going to get the 2 original strands of DNA, and 1 cell is going to get the new DNA. Semi-conservative, again, cell 1, cell 2, it says that each of them is going to get 1 of the original strands of DNA, and each of them is going to get a new strand of DNA. And lastly, for our dispersive, this shows us that 1 cell is going to get just chunks and pieces of the original DNA, and the other cell is just going to get chunks and pieces as well. But if you notice, the strands are not complete here. Each individual strand gets some of the old and some of the new, where the ones up here, you had 1 complete strand of old and 1 complete strand of new. So the dispersive was kind of like cutting the DNA apart and putting it together. So Messelson and Stahl did an experiment, and they were trying to conclude the semi-conservative model. So what they did here, guys, is they allowed the cells to reproduce, and they labeled the original DNA with a heavy nitrogen isotope. A nitrogen, another molecule that is, another atom that is going to be in DNA, and that's going to be in the nitrogenous base. So what they did was they labeled it with heavy DNA. And then they let the cells replicate and replicate. And then they looked at those individual cells, and they noticed that those cells each had some of the old DNA, which was heavier. They used nitrogen with an atomic mass of 15. And they noticed that some of them were heavier. And they also saw that they had some of regular nitrogen 14, which is like the regular nitrogen. They saw that they had some of that, which is lighter. When they centrifuged it, they had 2 different strands. The lower one, higher density, would be the nitrogen 15, and then the higher one would be the nitrogen 14. And since they had both areas, they knew that these cells had an old strand and a new strand. So that favored the semi-conservative model. Okay, so next thing, guys, we're going to go over DNA replication. And I know we talked about DNA replication during S phase of interphase when we were going into cell reproduction, but we didn't talk about how it happened. And that's what we're going to go over here. Whether you're a prokaryote or a eukaryote, there are a few differences between the way DNA replicates, but everything is pretty much the same. We are going to start with an area called the replication fork. So pretty much in this area, guys, our DNA is going to, it should be together, but our DNA is going to unravel, in other words, form a small bubble, and we call that the replication fork because on each side, the DNA kind of, well, it's like a fork in the road. But there's that right there, okay? So next time, guys, we're going to talk about this whole process of DNA replication. There's a good bit that goes into it, so I don't want to rush it today. So we'll talk about it next time, and have a good rest of your day, guys.